Let us begin by first taking 5.1 microliters of water. Then to that we're going to add 6.9 microliters of our digested beta-gal gene. With a new tip, I'm going to add 5 microliters of our digested vector, so the PET30A vector. Next, I'll add our 1 microliter of the T4 DNA ligase. Actually, I will add the 2 microliters of the reaction buffer first. It's normally a good idea to always add all your buffer components and I guess everything, like all the pieces of the reaction first. And the last thing you should always add is always your enzyme or whatever is going to catalyze the reaction. So now that I've added basically everything, the last thing I'm going to do is just add that T4 DNA ligase. So remember, because this is an enzyme, that is why I'm working with it in the ice. I want to try to make sure that it stays at ice as much as possible. So that is pretty much it for setting up the reaction. There's 2 ways we can go about the actual ligation. We can let it ligate overnight for about 4 degrees is usually, or 16 degrees overnight. We can also let it incubate at room temperature for 20 minutes. But remember, enzymes tend to work better at warmer temperatures, but they can also start degrading. So for us, we're just going to take the safe route. We're going to let it incubate at 16 degrees overnight, and this will ensure that we have the greatest ligation possible. So I will get back to you guys tomorrow. Alrighty, so it's now the next day, so our ligation reaction is now complete. So we can start with the next part of the experiment, which is the transformation. So all I'm going to do right now is just take 2 microliters of our ligation reaction. And add it to 50 microliters of competent cells. So it's very important that we keep these cells kind of like on ice and be very delicate with them. So I'm just going to add the reaction and just kind of flick the bottom of the tube pretty gently to let it mix and then put it right back on ice. So I'm going to let it sit in ice for about 30 minutes. I want to give it enough time so that the ligation reaction, so that our plasmid can kind of diffuse completely through the entire solution, just so that we can make sure we get a perfect transformation, or as close to perfect as possible. So I'm going to just let that sit, and then once the 30 minutes are up, we can start with the next step. Cells sit in ice for about 30 minutes, so now our plasmid should be properly diffused. So the next thing we need to do is actually heat shock it. So over here, I've prepped a hot water bath set to 42 degrees. You can kind of tell. So it's 42 degrees. And all I'm going to do is put in our competent cells and just make it, put it in there for about 10 seconds. So it's very important we go for 10 seconds, not too long, not too little. If the cells go in for too long, then we can actually cause them to rupture open and just break, so we lose all of our cells. But if we go too little, then when we heat shock the cells, the little pores that are going to come from the heat shock, they'll be too small for the plasmid to enter in. So I'm just going to start right now. All right, and then back into the ice it goes. So now I'm just going to let it kind of just chill in the ice for another 10 minutes before we can start with the next step. Our competent cells rest in the ice. The next thing we can do is start adding our 950 microliters of SOC media. So SOC media is a very, very nutritious type of media that's very perfect for, I guess, getting a really fast growth of cells, which is why we want to use this one. We want to give these competent cells as much nutrition as possible so that they can grow as quickly as possible, so that they are, I guess, super healthy. So once we've added the media, the cells are pretty much good to go now. So we can kind of just like tilt it back and forth to mix it. And now what we want to do is I'm going to take a bit of tape so that we can put it into a shaking incubator at 37 degrees. So here is a really big shaking incubator. And all I'm going to do is just tape our competent cells with the SOC media into the incubator, like so. And I'm going to incubate these for 1 hour at 37 degrees Celsius. Once these cells have been incubated for that hour, they'll then be ready to plate. So we'll get back to that when these are done. So there was a bit of a problem with the audio due to some fans kind of running in the background. I wasn't able to capture, I guess, very good audio when I was plating. So I just kind of want to re-explain what exactly I did here. So you can see that what I have there is an agar plate. And remember, this agar plate will have the antibiotic ampicillin. So we chose ampicillin because our plasmid has an antibiotic resistance gene specifically for ampicillin. So this means when I plate these cells onto the plate, we can expect that only cells that have our actual plasmid will be able to grow. So what I'm going to do right now is I'm just about to add 100 microliters of our cells to the middle of the plate. And once I add those cells, I'm going to open up a disposable spatula. So it's very important that this disposable spatula, or that the spatula is sterile when we spread these cells around, because we don't want to introduce any other type of bacteria that may be ampicillin resistant. So I'm just going to spread the cells all over the plate, and once I'm done, that's pretty much the end of the actual plating for the transformation experiment. All that's left to do is to throw this into an incubator at 37 degrees overnight and just wait for the colonies to grow up. And once they grow, then that is it.